Right, welcome back everybody to this third session of the BSMT conference.
Good afternoon. My name is Jonathan Edgeworth. I'm a microbiology consultant who works on the intensive care unit. And I also run a small diagnostics research laboratory at King's College London. So I'd like to thank the organisers, particularly Mark Wilkes and Brian Durden, the moderators for this session, and the British Society of Microbial Technology, for inviting me to give a talk about our experience with analysing secondary bacterial and fungal infections in COVID-19 patients. So first, to be clear what I'd like to focus on today, I'm going to restrict this to the intensive care unit. And I want to start by giving an overview of generally the pathogenesis, the epidemiology, the prevention of secondary infection, and take a view really on the state of infections in the UK NHS. I realise we have people from a number of other countries who will have different experience and rich experience. And clearly we look forward to hearing their perspectives throughout the conference. When it comes to the diagnostics, I'm going to talk about our experience introducing nanopore long-read sequencing technology, and focus on our experience relevant to the clinical diagnostic laboratory, and then looking ahead where we might go as we see hopefully the COVID pandemic coming under control. I would say we are a multidisciplinary team of discovery scientists, data scientists, bioinformaticians, healthcare scientists, including clinical scientists, biomedical scientists, research nurses, intensivists, and we all work together. Now, I spend most of my time on the intensive care unit. I am a clinician, but I'm embedded in this team. And so I'm going to cover some of the experience from my different colleagues' practice, right from the end-to-end pathway. Okay, so first let's talk about generally ICU-acquired infections. So patients coming onto the intensive care unit, they are covered with what we call primary potential surface pathogens. Relevant to the chest, that means in the back of their throat they have streptococcus pneumoniae, Haemophilus influenzae, perhaps Neisseria meningitis, all of which can cause infection but usually don't. You give someone an antibiotic, and these usually antibiotic-susceptible bugs tend to disappear. And you select for overgrowth of endogenous, predominantly gram-negative organisms in the gut. And then when the patient is laid flat and intubated, these bugs can come out either end and start to either colonize the respiratory tract that can lead to ventilator-associated pneumonia on the top, or they can colonize the skin. And when a vascular catheter is inserted, that can give access of the bacteria into the blood. Now at the bottom there, I've given a list of the dominant organisms causing both these infections. What's interesting is, for ICU-acquired infections, essentially it's the same spectrum of organisms the world over. People might have more of one than another, but it's usually the same list. The other thing that's interesting is that Staph aureus is probably the only one that's a common community pathogen, as well as being a common ICU-acquired pathogen. So now let's look at pathogenesis. Let's look at pathogenesis of ventilator-associated pneumonia or tracheobronchitis. This is probably the most important infection in the intensive care unit. So you've laid somebody flat and you've intubated them, and their stomach is full of bugs. Sadly, when they're flat, they're very apt to regurgitate gastric contents into their oropharynx, that's the B at the top. And then, because they have a balloon on the endotracheal tube that is inflated to touch the sides of the trachea, but it's not tight, when the patient coughs or gets moved, they can microaspirate bugs into their lungs, and that's what causes pneumonia. And for the last 20 years, there's been a concerted effort to prevent these infections on the intensive care unit. And they have what they call VAP bundles, ventilator bundles, where they elevate the head of the bed, put some chlorhexidine in the back of the throat to kill the bugs if they get there. Decrease the acid content in the stomach with stress ulcer prophylaxis, and then try not to too heavily sedate the patient and prevent deep vein thrombosis. Okay, now then, the second most important ICU-acquired infection is bloodstream infection. And these happen when the almost universal central line is inserted through the skin into a large vessel. And either at the point of insertion or during manipulation from healthcare workers, putting in intravenous fluids or just care, bacteria have contact with that plastic surface. They produce biofilms, they adhere to extracellular matrix proteins, and they get into the blood. Now, what happens next? Well, Staph aureus, it can go around the blood and get on a heart valve or in a bone, the top. Candida seems to go for the eye or very occasionally the heart. And so you go looking for Staph aureus and Candida disseminated infections. But to be honest, all the other bacteria, who knows where they go? Round about a few times into the black hole. As long as you remove the line there, then that bloodstream infection will stop. There is the same sort of catheter care bundle, equivalent focus as there has been on ventilators. And that's about how you put it in, cleaning the skin, how you care for it. There's chlorhexidine impregnated patches around the exit site and antimicrobial impregnated lines to reduce these infections. Now that's endogenous infections. Everything I've talked about thus far is endogenous. There's also exogenous infections. Now that's when patients come into the intensive care unit to colonize the resistant bugs. And those grow up even more when you give an antibiotic that isn't active against those resistant bugs. And then healthcare worker hands, you can see them there, touch one patient, and if they don't wash their hands enough, they touch another, and they transfer it to the respiratory tract or the skin. Now, in the UK, about 2% of patients coming to the ICU will have MRSA, about 9% will have an extended spectrum beta-lactam negative, about 0.2%, quite low, will have a carbapenemase producing Enterobacterialis. And I've written various names of the bugs down there. Some of those, as on the right, can become endemic and cause a real problem. The problem with microbiology testing, as we all know, is the delay in getting the results. So on the day when somebody gets an infection in the ICU and you want to treat them, perhaps within an hour, the golden hour, they're very severely septic, but sometimes you have a few more hours, but nevertheless that day you want to treat them. You can get laboratory markers from biochemistry and hematology, CRP, etc. You can send them down for a scan, see where the infection is, send samples for culture, but you have to give empiric antimicrobials, best guess. And it's not until day 3 that somebody will tell you what the results are. You'll be able to actually diagnose what the infection is. And quite frankly, in the UK, we often stop the antibiotics by day 5 or 7. So you've only got a couple of days to play with where microbiology makes any difference. In fact, it's often more for epidemiology, micro testing, you might say, than helping with the acute event. Now, what with the opportunity is with modern microbiology techniques is that they can use rapid molecular and genetic technologies to provide same-day results. Now, this is the holy grail of microbiology, I think it has to be said. The ability, on the day that somebody's unwell, to either say, look, there's no bugs in that sample, you don't need to give antibiotics, or this is the bug in that sample, and that's what it's susceptible to. You can give a narrow spectrum antibiotic than you were going to give. And then also, perhaps you can sequence genomes. You can very quickly say to somebody within a couple of days, there's been a transmission event between those two beds. So you can isolate, screen everyone, and call an outbreak meeting. Or perhaps once in a while, you say, I found something new. That's what SARS-CoV-2 was about, wasn't it? You can liaise with the national and international bodies quickly. Now, there's a lot of modern molecular technologies, but probably the long-read Oxford nanopore technology is one of the most interesting, the DNA and RNA sequencing. And this is really a sort of clinician's guide to it. These are pores in a lipid membrane, in a device the size of a mobile phone or smaller. And you process the sample, fluid, put it on the top side there, and these docking proteins go around, find the DNA and RNA in the sample, bring it to the pore, and then as it's unraveled and the DNA RNA goes through the membrane, generates an electric signal, and that can then be converted into a prediction of what the base is. Now the thing is, this is not as accurate as short-read technology such as Illumina, but it's what they call long-read. You can see there, it sequences it in one go rather than chopping into little pieces and having to reassemble. And then you can analyze in real time, and it's very relatively cheap to get started, unlike the money required for a large short-read sequencer. Now, introducing nanopore technologies, or indeed anything as complicated as this, is not simple. This is not like a MALDI, a new Vitek, moving from biochemical reactions, moving away from biochemical reactions. It's not even like introducing a multiplex PCR in some ways, where the clinical community has some understanding of what multiplex PCRs are about. This would require a wholesale change to the way the multidisciplinary team works. And so we've we've created a sort of a 3 or 4 phase program of evaluation, of discovery evaluation and pilot and analysis of the nanopore technologies. With the basic science over to the left there, with phase 1, more of the evaluation and developing reporting algorithms in phase 2, and then the pilot services where your clinicians make decisions based on the results you give. And I've written the names there of a whole host of people in our multidisciplinary team that started from the beginning, actually with the intensivists, help us take nanopore technology into service. The first test we decided to go for was what we thought would be relatively simple. And that is basically to provide what we hoped would be a same-day 16S service. Now, as probably most of you know, all bacteria have a 16S gene. We've got 9 variable regions, about 1,500 base pairs. And the conventional approach to providing 16S testing sequencing is to sequence a short read based on primers that cover perhaps the first 2 or 3 variable regions. And it takes, I don't know, 12 to 14 hours perhaps. That's what we suspected at the beginning. And it ends up being a test that spans 2 days usually. Whereas when we worked out at the start of this project for the nanopore, we could envisage the possibility with a stretch that long read sequencing provides you results within 1 day. So in September 28, we set about developing a 16S nanopore test for service. Now, it turned out to be much more complicated than we did. First of all, we had to make sure, we wanted to get this into an accredited service, that your nanopore 16S test could detect all bacteria. So we went back over the last couple of years in the microlab and found there's 167 different bacteria. So we had to either get them out of the lab, order them from ATCC, or for some of them just ask some DNA, and then go through and put all of them on a MinION to ensure that we could sequence those bacteria. And then when you use classifiers, either aligning them, programs to align them against known sequences, or what's called a KEMA-based classifier against a number of databases, you would accurately identify species level the bacteria. And after a lot of development of the bioinformatics pipeline, and I've shown the final pipeline up there where we actually use 2 classifiers, sequence match and supernatant, before giving a final pipeline report. 84% of bacterial isolates were correctly species, with only 16% misidentified. Now, when it came to the respiratory bugs, only Bordetella pertussis, which came out as Bordetella spur, was the only clinically relevant one that was missed. The bottom right there shows some very interesting results. Actually, we anticipated we'd need to sequence for 4 hours before the precision of the sequence going through the pipeline would be adequate to speciate. But actually on the left there, that's showing you the number of reads over 16 hours. The one on the right is showing the precision. And basically the precision after 1 hour is the same as for 16. So we only needed to sequence for 1 hour. Okay, that gave us confidence to move on to the phase 2, which instead of analyzing isolated bacterial colonies, we went to restored frozen respiratory samples, surplus to requirement, with ethics, after we'd thawed them. And to cut a long story short, over 100 samples, culture positive and culture negative, we saw concordance with culture in 75% of cases. Now, that was deemed adequate because they were frozen samples. Sometimes the differences could be due to issues with the with culture that's not perfect. Other times they were missing some upper respiratory tract flora, and that wasn't deemed clinically significant. And also importantly, that we identified 3 potential pathogens in the culture negative sample, giving you the realization that actually as a molecular test, you might be able to pick up bacteria that are missed from culture. We also identified predefined IQC criteria that could allow us to take this test into service. Now, we then also created a final report, a medical validation report for the 16S pipeline, and got the analysis down to 7 to 9 hours. I have to admit, that's not quite the picture I was going to show. It hasn't come quite out in the video. Actually, there it is now. Actually, that's more of it, showing how we brought the results together into a single final report. Okay, in fact, there it is now, the final validation report. Okay, so I'm now going to talk about the results from the pilot study. So having done phase 1, phase 2, December 2019, over 10 weeks in the intensive care unit, where they have an ECMO service, that's called extracorporeal membrane oxygenation. It's where the blood from the body is taken out, just like you do through a dialysis machine. And instead of removing all the urine as the this essentially oxygenates the blood and passes it around back into the body so you can rest the lungs. So these are severely unwell referral patients. We did 45 bronchoalveolar lavages from 37 patients, 15 who had suspected community pneumonia, 30 hospital pneumonia. And we compared this 16S sequencing service with all these other tests that were done, including a referral Illumina 16S, targeted PCR, atypical molecular PCR panel, and culture. And this is the summary results. We got concordance in 61% of cases for positive tests and 92% with the negatives. Now, we've submitted this for publication, and I think there's more detail here than I can go into to really tease apart what we found. But actually, where there wasn't concordance, not necessarily convinced that what was found was clinically significant. We had very fixed, predefined criteria for developing the test. So what I want to do now is to just give you some vignettes of what happened to individual patients. So this is a 48-year-old woman, came in for ECMO, who's been in for a while. We think it's an infection that brought her in, not quite sure. You can see from the CRP markers that they initially came down, but then increased. And around about day 14 into admission, she got a ventilator-associated pneumonia. We took a BAL for 16S. We did the 1-hour sequencing test, 1-hour sequencing. The sample arrived in the lab at 9 a.m. We rephoned the results at 5:30 p.m. If you look at the bottom sequence match of classified reads, there is a whole host of bugs in there, as you might expect. Technical validation, we said that was E. coli dominant with Staph aureus, plus mixed anaerobes and upper respiratory flora. We advised tazocine that day. Subsequently, over the next 2 or 3 days, E. coli was grown, found to be fully sensitive, completed the tazocine 5-day course, patient off ECMO. Second patient, 43-year-old male, polytrauma, ingestions of multiple substances. Went across the road, damaged his lungs, possible aspiration, comes into hospital on ECMO. Only been in hospital about a day and a half, 2 days. And he's on tazocine. And we send the BAL off for 16S. And this is what we find, is that the sequence predominantly is of Haemophilus influenzae. And so this looks like an aspiration, Haemophilus mixed anaerobes, and we advise to de-escalate from tazocine to comoxiclav. Now, what's interesting is as the culture results come out over the next 2 to 3 days, actually culture grows scanty E. coli. When we went back to look at the sequmatch bioinformatics output, we found 12 E. coli reads, only 0.01% of classified reads, and that's why it was discounted. But actually, that was the bug that came out in culture. And it reminds you, scanty E. coli, 3 colonies on a plate, that's very sensitive. It's unlikely, probably, that 16S is going to be more sensitive than culture. It's about the speed. There's also here, you know, it's scanty E. coli, you might say, well, it was an E. coli pneumonia partially treated. But no, it probably wasn't. That's emerging. That's probably not the cause of the infection. The infection was Haemophilus influenzae. Okay, a third patient, 48-year-old woman with desquamative interstitial pneumonitis. Been on ECMO for a while, heavily immunosuppressed with cyclophosphamide, and developed some ventilator-associated pneumonia. Didn't have any help from previous microbes, CRP 260. Sample arrives at 10 a.m. in the lab, and we phone the result back at 6:30 p.m. That's the sequencing report. Lots of different bugs. Call that Pseudomonas aeruginosa, strep Milleri group, some mixed anaerobes and upper respiratory tract flora. Bottom line, tazocine. Patient had temperature 38 and CRP was 158. Took them off ECMO actually around the time of giving them tazocine, and the inflammatory markers reduced and they were doing well. But look at this. 2 to 3 days later, what did we have? Pseudomonas aeruginosa. So that's what we got in the 16 days. But it was resistant to tazocine. However, the patient had got better. So we'd given ostensibly the wrong antibiotic, the patient got better. We did a repeat NBL before changing to ciprofloxacin, and actually the Pseudomonas had gone away. I think what this illustrates, I think those who work on ICU know, is there is not a good correlation between the clinical state, the bacteria you grow, their susceptibilities, and the antibiotics you use. That's the sort of uncertain, heterogeneous environment that you work on in ICU. Now, when I reflect on the nanopore service, a number of conclusions I'll come out with. The clinicians loved it. They ended up selecting the patients they could actually manage a result at the end of the day. And they became more interested in the patients who didn't have that, but they wanted to exclude bacteria to increase immunosuppression. We need to adapt the test to optimize it for that. Interestingly, they said the main clinical unmet need at that point was, what about fungi? Not what's the antimicrobial susceptibility. From the lab side, we could only do few a day, 1 to 3. That's not going. We need to scale up. It's also significant biomedical scientist hands-on time that increases the unit cost. We had a few failed tests due to bioinformatic issues and a couple where the pore count dropped on the Fungal and loading issues. But by and large, I think probably about 90 to 95 the test went through that day. It was ready to go for ISO 159 accreditation. And then the COVID pandemic arrived. And of course, it all hit us hard. So March 2020, we stopped the 16S test. And this is what it looked like for us, and similar, I'm sure, to people around the country and around the world. We've had 2 waves of admissions. This is the number of PCR positive tests in our laboratory. Orange on admission, green nosocomial acquisitions. And schematically, we're showing what happened to our ICU. Essentially, it had to expand massively. So a 15-bedded ICU, 2 patients per pendant, doubling up, a foot between the beds. And there's so many reasons why secondary infection could be more in COVID patients. They're prone to more steroids, they get lung destruction from pulmonary emboli, infarctions and abscesses and pneumothoraces, fistula, and they end up on the. And then the opportunity for multi-drug resistant organisms to spread is much higher, given the fact that you're bumping into each other around the beds. You're wearing multiple layers of PPE. Those are just all right for the transmission of organisms. So what about what happens to the epidemiology of the bugs on these intensive care units? So what I would say is, as you would predict, look, it's going to be the same bugs the world over that we traditionally see. You're not going to suddenly see, I would suggest, a whole bunch of different secondary infections in patients. You're just going to see more of them. In fact, that's what's found. So this is a multi-site UK and Italian study, and probably, it's reasonable to say, within a couple of weeks, 40% of patients will have some kind of hospital-acquired infection. Pneumonias and bloodstream infections. On the left is the usual bugs. Now, something else happened to us when we first got hit by that first pandemic wave. A postdoc arrived from Justin O'Grady's lab, called Themis, who just published this paper in Nature Biotechnology on nanopore metagenomics and how that could help with rapid clinical diagnosis. Now, the difference with metagenomics is you don't PCR up the gene sequence. You essentially directly sequence all the DNA and RNA in the sample in real time. Now, you can't freeze samples. You've got to be fresh samples, or else you'll just get overwhelmed with human reads, human DNA that needs to be depleted before you can go about sequencing the pathogen DNA. So basically, all of the phase 1 basic science had been done by Themis. She arrived. A week into the pandemic, we say to her, well, can you do some metagenomics on respiratory patients? And she can, and so off we went. So this gives an idea of what happened on the ICU in those 2 waves. Wave 1 for us, we had 300 mechanically ventilated patients, peak occupancy of 113 patients. In wave 2, we had 440 patients with a peak occupancy of 155 mechanically ventilated patients. And there was more than that in the intensive care unit, perhaps up to about 230, 240. The others were not ventilated. And the top there shows you how it wasn't just our usual ICUs used. The pediatric ICU, the overnight intensive recovery, a chronic respiratory unit, a cardiovascular ward. And hospitals around the world will have to put on similar things to look after these patients. So this is an overview of the respiratory culture results from respiratory specimens in wave 1 from our mechanical COVID-19. Just like all the other studies, Klebsiella species up there, the dominant organism. Now, admittedly, not all, it's Klebsiella pneumoniae and Klebsiella aerogenes, and a few years ago, it was Klebsiella aerogenes was called Enterobacter aerogenes. And so the reason why Klebsiella seems to be everywhere at the moment is in part because of the name change. Nevertheless, Klebsiella pneumoniae did stand out as the most dominant organism. And 63% in our study had some kind of acquired resistance during that patient's stay. And interestingly, the bugs, the Klebsiellas with the acquired resistance were more likely to get into the blood and cause bloodstream infections. In fact, 3 times more likely than susceptible Klebsiella pneumoniae, and also wild-type Klebsiella. So that's the microbiology. What did we do? Well, we couldn't introduce a pilot service to guide therapy because we hadn't done an evaluation yet. What we did do was take 43 fresh respiratory specimens using the ethics and the pathways developed over the 16S study from 34 patients and conducted metagenomic sequencing. Now, a lot of work has had to be done on the pipeline, and this is just illustrating how you have to think about identifying the bugs. You have to do 2 hours sequencing, by the way, we found with metagenomics, both to identify the bugs and do the resistance prediction. And this is the sort of results that we have seen. So 4 samples on the top, remove the human DNA. These are the classified reads, and shows you, between 1,000 and 100,000 classified reads. Very clear identification of the organisms, including here Aspergillus fumigatus, 77 reads in that sample. The bottom panel, just showing 2, is the gene detection of the antimicrobial resistance genes and how we correlated that with phenotype. And this worked for most bugs, but not some like Acinetobacter. So we haven't yet been able to identify AMR genes. Now, the challenge, the opportunity, is to turn that 2-hour sequence result within a 7-hour workflows into a same-day actionable antibiotic treatment decision. And this is not a complete table, but all I'm really trying to illustrate here is that the matching between genotype and phenotype is really good, although, third one down, S34, we found here additional genotypic resistance that was not phenotypically identified. So that would have led us to prescribing meropenem probably, whereas you could have got away with a standard. And then S37, sometimes you have a mixture of organisms that you have to contend with. So that's just giving an illustration of how you turn the identification and genotypic resistance into an antibiotic treatment. I just want to say a little bit about 2 slides about invasive aspergillosis. I'm not a fungal expert. All I would say is that we know aspergillus is ubiquitous in the air. If you have a look at people's respiratory microbiome, you don't usually see aspergillus. I understand in healthy individuals you have other respiratory fungi, but you can see it in people with chronic disease. Now, most COVID patients were otherwise healthy. They may have had some predisposing hypertension, hyperlipidemia, and etc., but obesity, but they didn't have chronic lung conditions by and large. So most of them on the bottom there will have not come in with aspergillus. Now, they might pick up a bit of aspergillus in the ICU, although they're on closed ventilation. So that's probably generally in most settings unlikely. But there will be a few that come in on the top there. And when you give them steroids in the context of SARS-CoV and intubate them, you'll end up with a focus of invasive aspergillosis. But in the post-mortem cases that we did, turned out to be small nodules that weren't actually the cause of death. Although I think in some patients with more steroids might change that view. Going back to the overall microbiology results from the wave 1, we cultured aspergillus from 6 patients, so actually it was very low for us, 2%. Now, we did bronchoalveolar lavage galactomanans that's often used in the diagnosis of aspergillus in 51 patients and found a further 2 more. And on the bottom here, I'm just showing the variation of reads of Aspergillus fumigatus in the 4 culture samples that we had in our metagenomics data set. Go from 16 reads to 2,500 reads. Now, this is quite interesting. There is a long way to go. What do we currently use? Galactomannan, so detecting a biomarker. PCR, a band on a gel. I wonder just how much extra confidence one will be able to put in a sequence report in terms of interpreting that as presence of Aspergillus fumigatus, and how many reads you need to call that a positive result. Okay, so I've just got a few more slides now, just to point out a couple more interesting things about being able to do nanopore sequencing. That is that if you continue the sequencing overnight and come back in the following morning, you have adequate sequence to align the genomes and do SNP typing. And on the left here, we had 7 samples with Klebsiella pneumoniae in them. And in most of the Klebsiella pneumoniae, they differed by over 10,000 bases. They clearly are unrelated. But look, we had 2 here that only differed by 3. When we then compared those 2 with 2 blood culture isolates from 2 other patients, this was actually a Klebsiella ST307, ESBL, and multi-drug resistant. All of them only differed by a small number of SNPs, and so were probably related. And the epidemiology in the bottom there of the ward stay overlaps confirmed that. Interestingly, we also identified an unsuspected outbreak of Corynebacterium striatum. So again, on the left, I've shown this a few times. This is all our respiratory culture results on the patients. And from our metagenomics patients, 8 of them, 24%, we grew Corynebacterium striatum. Now, often in the laboratory, probably you would just ignore that and wouldn't report it, but often these were in pure culture. When we aligned their genomes, we got 5 samples from 3 patients. They differ by only around 100 SNPs. And in fact, the inter-isolate SNP difference was the same in the 3 samples from 1 patient as between patients. So this was consistent with there being a Corynebacterium striatum outbreak. When we aligned all of the patients who had cultured the striatum, it was restricted to 2 wards where we think there was some transmission of Corynebacterium. Okay, so same day ID and sends, and the next day you can point out where there's a transmission. So this looks technically feasible, something that's worth pursuing to take into service. But what problems is it? How will it help? And I think we need to this will be different for different ICUs. And I think we need to work out just what the burden of infection is. And the points that I would make, first of all, are that we used to have a lot more infections on intensive care unit than we do now because of those preventative studies. This is data from every patient admitted into every UK ICU over a 10-year period up to 2016. This is a population level study. And what it showed was that ICU-acquired bloodstream infections over that time period decreased by about 80%. And that was around the time when MRSA and C. diff reduced by 95%. So the UK has seen some extraordinarily positive results in reduction in ICU infections. Then when you think about outbreaks, the literature is full of reports of outbreaks, but on an individual ICU, how frequently are they? Now, this was an old study we published 2013, where we looked at all the bacteria from all the samples on every patient in the ICU over 9 years, and then looked using a WHO stat scan of how often there was predicted to be some clusters of outbreaks. And broadly, mostly, we saw with the red and blue short, self-limiting clusters of outbreaks of organisms. You also have to look at the burden of resistance and the clustering of resistance and link that with the use of antibiotics. It can become quite a data analytic challenge. And I think something interesting we saw here is that over this 9-year period, there was 5 periods of time where there was clustering of the antimicrobial resistance determinants for different antibiotics and different species, just one dotted red box there. That's perhaps indicative of transmission of mobile genetic resistance elements as much as transmission of the organisms. So I'm just giving a pointer there about what we yet don't know about transmission and where we might be that can help us position appropriately metagenomics. Okay, so this is my last slide, and I think I'm just about within the half hour. What conclusions have we got from the work we've done over the last year or so? So I think there's still a lot to know about the epidemiology of secondary infections in COVID-19 patients. But I think broadly, particularly when you've got an inundated intensive care unit, there's probably more infections and more resistance. And that'll be a challenge for everybody as the pandemic hopefully comes under control to go back to baseline. I think there's emerging evidence that 2 bugs are dominating. One of them is Klebsiella, particularly in the ventilator-associated pneumonia. And actually, I haven't talked about it today, but probably Enterococcus faecium is a cause of bloodstream infections. It is the second emerging organism. But of course, this pandemic is continually evolving. Things might change with changes in host immunity, new treatments, emerging variants. I think the work that we've done and other studies, and actually I should apologize here, I haven't covered the literature, I haven't looked at all the excellent work that's been done on other studies. I felt in half an hour probably the best I could probably offer is to go through the work we've done. I think you can see we're taking a step back, that there really is potential for long-read sequencing to impact on a number of really important priorities on intensive care and the wider healthcare system. You ask me what the challenges will be. I don't think it's in the technology. I think the technology is going to be there. There's lots to do, but I think we'll get there. Providing evidence of clinical utility and the cost effectiveness, and then moving from this proof of concept pilot service stage into embedding it in accredited diagnostic laboratories as a service for the whole of the NHS, the whole data science and technology challenge. And then finally, I would say the whole multidisciplinary team, from the discovery, healthcare, behavioural and data scientists, need to work with doctors and nurses, public health, infection control and pharmacy to get this in. they all have to change their practice. I mean, if the doctors just say, really nice result, I'm not going to change it, my practice, carry on the same antibiotics. If the infection control team just say, tell them to wash their hands more, or if public health just say, well, that's interesting, let's wait till we get more data. If people don't do things differently themselves all across this spectrum, we could end up wasting money, and that's our challenge. And we wrote this piece at the bottom about how you dissolve boundaries between professions and specialties to try and help translational research, so you can get this team ethos together, get novel technologies into service. We felt that was one of the main challenges and one of the main opportunities that COVID-19 gave to bring us all together. So thank you. I'm going to stop there. Thank you for listening, and I do look forward to hearing the next talk and contributing to the discussion later. Thank you.
Thank you very much for that presentation. A vast amount of material and some quite fascinating stuff in there. Sorry there was a hiccup at the beginning. I hope everybody got the beginning of your talk, even though you weren't actually introduced. So sorry about the hiccup at the beginning, and hope that you didn't get a proper introduction, but I think everybody's worked that out, that you from the program.
Yes.
Vast amount of material there. And I think at the end you were saying the difficulty is getting people to change their practice and to change their ideas in the light of this. It did strike me that one of the things you were finding on the positive side was identifying perhaps underlying infections when traditional culture shows the things that are easy to grow and easy to report. And yet you were able to pull things out from underneath there that might have been more significant.
Yes, can you hear me, by the way? Yes.
I can.
Okay, so you're absolutely right. The 16S particularly showed you a whole bunch of different organisms, and sometimes it would be like Haemophilus influenzae that was probably the cause, rather than something scanty of another bug that hadn't been killed that might have been emerging. Interesting, one we didn't show was we had somebody with influenza and group A streptococcus, and that was just 40,000 reads, group A streptococcus, and absolutely no other bug. So there are some pure infections when you see absolutely nothing else. And then, of course, Corynebacterium striatum, there are outbreaks of Corynebacterium striatum. Might it be that there are some organisms that spread quite well, but because they cause no bother, we don't care about them? But they could be a marker, they could be a quite frequent event we can use to assess the impact of interventions. But yes, you're right. That opportunity to learn, find out much more what's going on in the microbiome beyond culture.
Thank you. Yes, we had a question quite early on for you, which was that somebody saw that you had CRP and PCT on one side of one of your slides as markers. And the question is, do you think there's still any mileage in them, or have they been around too long before showing and really are outdated, or have they been around all this time and can only now show their worth? Or is all this to be superseded by the direct sequencing, the molecular approaches that we have now?
Yeah. So there's huge uncertainty on the intensive care unit. We don't actually know often who has infection and who doesn't. And we know the consequences potentially are bad. And there's lots of people there who have an interest in that patient, the intensivists, perhaps the hematologists, the specialists, the pharmacists. And so I think people in that environment of uncertainty will try to bring together any piece of information to help them make a decision. And we look at CRPs, we get used to looking at them. I think after you've done it for a few years, you know their shortcomings. What's interesting is not a lot of hospitals, at least in the UK, can get procalcitonin, PCT. It's more expensive. And they really want it because we, like everybody, we have uncertainty, and they hear about PCT perhaps being specific for bacteria, and so they want it. So we've been fortunate, we get it frequently, but I would say, is it a game changer? Well, to be perfectly honest, in the intensive care unit, not. We see some people who clearly got a bacteremia, PCT is less than 0.5. Others, the PCT is 100, but actually they've got an infarcted kidney after an abdominal aortic aneurysm. So these biomarkers, none of them are perfect, but actually in this environment of uncertainty, they're helpful. The second point I'd make is, I got looking at the bugs, but the intensivists look at the patient. They care about sepsis. We didn't talk about sepsis today. So if I come along and tell them what the bugs are for sure, they'll say, yeah, but hang on a minute, is the patient septic? But I want the temperature, the blood pressure, the respiratory rate. Do they have a shiver and a shake? What's the CRP? They're all looking at different things. I don't think they will easily get rid of anything for quite a while.
Thank you. Completely different here, but we had again an early question in pointing to the Asia experience with mucormycosis, described here as the black fungus. How has this happened? Do we know why it's happened? Is there any equivalent outside of Asia?
Yes. So you know what? When I was drawing that picture with the aspergillus, with the black fungus in mind, because I sort of wanted to say, if there are patients in India who are exposed to mucor in the environment, perhaps in a rural setting, I don't know, some of them may have got a small amount of the mold in the back of their throat, or they might have inhaled some into their trachea. When you give them steroids and intubate them, that's what's around. So that's what will grow up. So I think the key difference will be the exposure. And if you go around the world, in different countries, there will always be different community bugs that might just have got in there. My comment about the ICU was, once you've got into the ICU and you've got rid of your community bugs, that's when I think everything's the same. But in terms of bringing in, each country may have its own specific organisms. And it sounds like mucor is something that's being identified, albeit rarely, I think it's fair to say, in India. But please correct me if I'm wrong.
I think they were talking about very different environmental situations.
Yes.
In which the COVID infection is being superimposed. Yes.
Yes.
Again, we've had a question here. It says, is there a good way of correlating antimicrobial resistance detected by the sequencing with the bacteria that you ID by the sequencing? Do you link the resistance directly to the organism identification, or do you have to work through that case by case and try and match them up? Is there a straightforward link between what you find?
So get the quick question out of the way. Yes, you can correlate quite well enough, but would always like to get better. The genotypic detection of resistance determinants with your phenotypic testing with DISCs or Vitek, that's fine. If you're asking me, what do you do about a mixture of organisms where you identify these resistance elements and it's only 2 hours sequencing, you haven't been able to align the genomes, how do you know which resistance gene is in which of those two bugs? Say you've got a serratia and a Klebsiella in the same sample. Well, that's what our bioinformatics team are working on. I don't know where they've got to. Maybe that Nick Lohman can answer that question a bit later. I think that's something that's in hand, but there may be some residual challenges clearly in a mixed sample.
That's because that's clearly you were interested in both aspects of that, both the organism and the resistance. Yeah. We do need to be able to match them up, don't we?
Yes, we do, exactly. Although in some ways you could say if the resistance determinant is there, that's enough to make you say, I don't care which bug it's in, I'm going to use that to guide my antibiotic. But that's at one level all.
It is. It's just that, again, that also comes back to the quantitative aspects, doesn't it? In that you're looking perhaps for more predominant organisms to put the greatest weight on them, and that's where you need to be able to match up the antimicrobial resistance with the organism that you're finding.
Yes. Although, can I just say on the quantity that one thing I've been quite impressed by with the metagenomics, particularly with the cutoffs that are used, is it didn't go identifying with the settings that we used a whole bunch of bugs that we don't quite know what they're doing. Yes, there was the metagenome that had to be got rid of, but the bugs that were called were by and large the ones that were in the culture. Might have one or two here and there, but it wasn't a wholesale uninterpretable mess. That was a big concern, that suddenly there'd be more info than you could deal with. And the 16S showed that. I think we hadn't thought through why we had so many more bugs in the 16S than the than in the metagenomics. It might be that the COVID patients were quite pure in some ways, relatively healthy, perhaps didn't have not many chronic diseases, etc. And it was whereas a lot of the other ones might have been aspiration, more complex submissions. That's something we have to get to.
Yeah. Now, a question here, which is a more general one rather than the specifics. It says, what shall we do to speed up the solution for the delay getting new diagnostics in the NHS accredited for use in the laboratory and for clinical use? There is a lag in the uptake, isn't there?
Yes.
There always has
I think, yeah, I think I was trying to prompt that by showing how we worked and showing that publication at the end. You know, in a sense, it feels like sometimes that we have a methodology, phase 1, phase 2, phase 3 clinical trials for getting drugs into service and into practice, and that works. And there might be an attempt to fit diagnostics through the same pipeline, but it might need its own bespoke steps. And particularly, you know, do the science, do the evaluation and go into a pilot service, get it embedded and see how behavior changes. And once you've shown people what can happen, that's what then brings to life the possibilities. So we feel it is getting as fast as possible into the point where safely you can start delivering a new technology and start to see what happens. And it's about getting different professions and specialists together to work on the problem from the start. What you mustn't have is handovers where somebody spends a year developing something and then walks into a room and says, hello, look what I've discovered, you've got to use this. Or walked into intensive care and saying, you're going to love what we've got. Have a go, you know, start requesting this. Have them in the room at the very beginning so they create the solution around the clinical need, and so they're pulling it in because it's theirs as much as ours. I think this is about behaviours and about teams, to be honest.
I think that's certainly a case that changing behaviour is the key to it. It's a professional behaviour that's ingrained over years.
Yes, yes.
As we look forward in this, can you actually envisage that we really do change our practice to this, and that we can get this same day diagnosis into key bacterial infections and they get the right treatment going? Because that clearly, as you think you said in your talk, that is the real goal of this, to get treatment focused.
Yeah. So I think we will, up to a point, beyond which I think it's too uncertain to know. So our objective for winter is to pilot metagenomics in the same way we did for 16S. So we're providing it every day to the ECMO units across, or to, you know, if we do, God forbid, have more COVID patients or flu patients, and get some specialist units to get used to it and to publish that work to sort of help generate a conversation. And then you could envisage it being in a network situation where you bring together same day ID and SENS, outbreak detection, and perhaps a sort of sentinel rare and unknown capability, so that you've got enough samples each day. You need to have you can't do 1 on Monday, 3 on Tuesday, nothing on Wednesday and Thursday. You know, you've got to each day you've got to have a lot of work so the unit cost is down, so that you can drive things forward. And then some kind of evaluation of utility and cost effectiveness. Now, the thing I don't know is when you get to that stage, you say, yes, it is possible, but it's still an awful lot of effort. And actually, all our microbiology laboratories are in a hub somewhere 2 miles down the road. And, you know, the logistics, getting the samples there is too long. And you might run out of energy. You need an awful lot of energy to keep overcoming different perspectives and views and rationales. And it's just whether, probably at multiple levels, from commissioning downwards, there's a desire to really see this through to embedding in a long enough period of time so that we can overcome some challenges that we don't even know exist yet, to realise that benefit. I think there's a good chance COVID has probably been the biggest momentum enabler for this to happen that we've felt, and I think that might continue. There's a lot of gridirons around the country now, I understand. So let's see.
One thing I wasn't very clear on about perhaps earlier in your talk was just how much was it, how much secondary infection that has, respiratory infection that has been in the COVID patients. There's been so little publicity on this generally. I know it's different amongst the profession, but generally it's all been about the virus and its effects.
Yes.
I'd be struggling to work out just how much the secondary infections have impacted on the patient. It clearly should be really important. It's been difficult to just see how big that problem has been.
Yes. So there's only two main secondary sites, chest and blood, as far as I can tell. So they don't have many operations and the urine's fine, and they're usually relatively first-time pass-through hospital. So chest, very difficult to ventilate, very chronic, difficult lungs. They will, you know, the majority will be colonized with bugs if you keep them on the intensive care unit long enough, 3-4 weeks. 40% by 2 weeks. I think that's been, yeah, half the patients have had some kind of hospital-acquired infection in 2 weeks. I think that's a pretty good ballpark figure. And it's your usual bugs. One thing I would say is they have a broader range of bugs. So normally a patient's a Serratia patient for their stay, or a Klebsiella patient. These had Enterobacter, Serratia, and Morganella one day. The next week it's Pseudomonas and Acinetobacter and Stenotrophomonas. Multiple bugs frequently throughout. When it comes to bloodstream infections, we're just submitting this at the moment. Wave 1, we saw a lot more bloodstream infections, but the rate per 1,000 bed days was the same as background. Wave 2, we saw an increase in some bacteria, Candida and Enterococcus faecium, and so the bloodstream rate has increased. And we wonder whether that's an earlier use of more steroids that's disorganizing the microbiome, leading to the faecium bacteremia, because they're not coming from the chest. We think they're primary endogenous. So I think the bottom line is, actually, because everybody coming into the intensive care unit gets a line, and lines cause bloodstream infections. If you carry on with your general practice, the rate will be the same. And then there might be other phenomena such as what are steroids doing?
You have two organisms that traditionally have been associated with steroid use. Traditionally over 7 years, and to see them coming through should not be unexpected. It's interesting to know. I'm just checking to see if we've had any more questions through.
Well, I suppose I would say is, well, the only thing I would say is I don't think they're going to beat the multiplex PCR for atypical respiratory bacteria. They're very quick, aren't they? They're excellent, very sensitive, and in fact, the sensitivity is what you need. So I'm not sure that metagenomics necessarily will be able to beat that atypical PCR. In fact, we missed a case of Chlamydia in one of our patients because the primers for the 60 nest didn't pick it up, which was interesting. So and of course the PCR, the multiplex PCRs as well. So it is possible that for the community, multiplex PCR admission and the atypicals will carry on doing that, and the metagenomics might be more for the secondary infections in hospital, but that's just a broad signpost.
I've got something rather long that's coming through. I think it's a statement rather than a question that says the speaker's spot on with problems of matching up multiplex PCR results along with the PCR detection of resistance genes. Then about some companies such as Microgen offering a test for urine analysis for UTI, and the patient then gets those results. But it needs then clinical advice. I'm working through this. So I'm not sure it's a question. I'm looking to see if there is a question. And I think it's really the need for the interpretation of these results, which I think you've covered. And it does need the medical advice as well as the basic. I think the point is this is making is that as well as the scientific result from molecular analyses, you do need the clinical interpretation and advice on what it means and how to do it. I think that's been your main thrust throughout all of this.
There needs to be a conversation, and it's about the interaction between pathogens and hosts. So you need the intensivist there who can talk about the patient, and then you need somebody who can give an infectious diseases diagnostic microbiology perspective, and you just need to have a conversation and reach an agreement about what to do, so everybody sleeps well at night and you're comfortable, you've done your best. Nothing like waking up in the middle of the night and thinking, oh gosh, perhaps I should have done different. Am I sure I'm right? Because this is very difficult environments.
I think that's a very good point to end on. Thank you very much for your talk. As I'm sure everybody picked up, your pedigree as an infectious diseases clinician and intensive care specialist, bringing all that together and linking across to the most up-to-date molecular diagnostics. Thank you very much indeed for that. We will now have a few minutes break before we start off again at 3 o'clock. Thanks, Jonathan.
Thank you, Brian.